Benokranı kişileri teşekkür ederim başlama istiyorum. Ondan nazik davetiniz için mükemmel atölyesi ve silginden dün geçir. Okay, I have no idea what I just said. But I hope Google Translate got it right. But I'd especially like to thank you all for coming out on your Sunday. And Başak also for all of her wonderful take care of me. So, when we talk about predictive health, which is a big part of the discussion in America now, I think one thing we have to realize is that the National Institutes of Health are actually about disease. And I think in the West our attitude is that we wait until we get sick and then we go to a doctor and the doctor tells us what we need to do to get better. And there's a different culture that has been in the world for thousands of years which says that we should think about our health behaviors before we get sick and behave in ways that prevent that from happening. And so the question I'm interested in is whether or not we can use personalized genomics to help each of us make those health decisions. So that instead of genomics being used for a few percent of the population who have debilitating diseases at birth or who have cancers, that maybe it's something that will eventually become a part of all of our medical care. And this, of course, is in the future. I mean, right now there are more important concerns than just providing medical care for everyone. But I want to talk a little bit about one of the directions we're taking. And the mentor for me is this man here, Ken Brigham, who set up the cohort that I'm going to talk to you about in Atlanta. And one of the quotes in this book is this: as many of us as possible should age with grace and die with painless dignity of natural causes. So it's not just about living longer, but it's about living healthier. And so wellness and health are what I'm interested in. Okay, so how can we use genomics to enhance our own understanding? Does anyone here use the company called 23andMe? No one? Has anyone heard of this company, 23andMe? One person. So I've got 23andMe too. So what this company will do is for about $100 US, will provide all of your genome, your genotypes, not the sequence, but genotypes throughout the genome. And then they provide predictions. So in my case, for example, they tell me that I am 4% Neanderthal. They tell me that I'm high at risk of heroin addiction, which is not true. I don't even know where to get heroin, so I'm at zero risk. But if I did, then I'd be at high risk, apparently. More seriously, what they think they can do is predict disease. And this is how it works. So the idea is you have a condition like hypertriglyceridemia. So high triglycerides, high fat levels in your blood. And you can measure what the prevalence is in the general population. Okay? So everyone in this room is happy, yes? We're all healthy and happy, yes? Yes, okay. Come back 20 years from now, will we all be healthy and happy? Look around you. Someone next to you will have diabetes. Someone will have cancer. Someone will be having, well, hopefully not early Alzheimer's disease, possibly. Okay? So all of us are going to get these common complex diseases. And so the question is, can we predict that? So we can measure what the prevalence of each of these diseases is in today's society. So if you look at type 2 diabetes, in America, it's about 15% of all adults over the age of 50 have diabetes. For coronary artery disease, it's actually as many as 50% of the American population. Whereas for schizophrenia, it is just 1%. So now the question is, can we take that population prevalence and modify it by our genomic information? All right? So the idea is that people doing genome-wide association studies, or GWAS, are finding hundreds of genes which contribute to each of these diseases. This morning we heard about single genes responsible for malaria, thalassemia, for example. All of these diseases are much, much more complicated. They're an interaction between genotypes and the environment. For height, for example, we now know 200 genes. This has been measured in 250,000 people. They have found 200 genes which explain 10% of height. If you took those genes away, we would not notice it. We'd not notice the difference. Okay? And yet, that's the state of the art of genetics. So the idea is that we can do that for all of these diseases and find the hundreds of variants that contribute to cancer and diabetes and schizophrenia. So what you do is you take the baseline risk and you modify it by each genotype. So this genotype increases the risk for this person by maybe 10%. This one has no effect. This one increases it. This one has very little effect. Maybe the next one decreases it. So you go through that process for each of these genotypes and you come up with a modified risk prediction. So for this person, they're at slightly increased risk of coronary artery disease relative to everyone else. For obesity, they're also at higher risk. But for hypertension or high blood pressure, they're at lower risk. Okay? So we can do that for all of these diseases. And we can debate about how useful that is. But as the science gets better, we'll get better and better at that. And then what we need to do is to convert these, what we call riskograms. So this is from actually Atul Booth's group at Stanford, Ashley Altman et al. So what they argue is that in fact all of these diseases interact with each other. So if you are obese, that affects your risk of diabetes and coronary artery disease. So we can estimate this, and then we put them all together as well with our environmental exposures. So what drugs do we take? Have we got a history of disease? Do we exercise? What's our diet? Are we doing things to reduce our psychological stress? That's the grand aim, is to give each person one of these plots which highlights for them where their genomic and environmental and clinical exposures are. And the question I am asking in my group is, whether or not we can actually add functional genomic information into this. Can we use gene expression profiling, for example, to improve on this classification? So I'm going to, am I speaking too quickly? Can you follow? Okay. So I'll try to go a little bit more slowly than I normally do, so that you can follow. And so I'm not sure how much I'll get through, but follow along. So let's start with the challenges. Okay? So the first thing is to develop a genomic or a genetic predictor. So for $100, I can generate 500,000 genotypes throughout each of your genomes. And then we can actually use those to predict each of these diseases, to establish where each of us is relative to everyone else. Now the immediate problem is that none of these predictors explain more than 25% of the risk. In fact, most of them are more like 5% of the risk. So how useful can something be if it only explains that small amount of the variation? Okay? It will get better. We've been doing this for 4 years now. 10 years from now, I suspect that we'll be much, much better. Secondly, we have to optimize that risk score for each individual population. And we have to optimize it for the way we do statistics. A very important concern is population stratification. So if a disease is at a different prevalence in Turkish people than it is in, say, English people, and the genotype's different in Turkish people than English people, then you can get a false positive association between the genotype and the disease. Okay? So we have to adjust for that. So all of this has to be done on a population by population basis. And so I would hope actually that eventually there will be this sort of genomic population genomic project for Turkey. This is all discovery genetics. This is done in sample sizes of 50 to 100,000 cases and the same number of controls. It's enormous. and very expensive. Probably $1 billion has been spent on this in the last 5 years, or one fighter jet equivalent. Okay? The problem from there is to go to N=1 genetics. That is personalized genomics. Okay? How do we convert these discoveries in understanding your own personal risks? Okay? So can we include functional genomic data? How do we incorporate the environment? How do we incorporate rare variants and de novo new mutations that are unique to each individual person? These are all of the challenges. And then we have to establish that it's useful from a clinical perspective, which may be the hardest challenge of all. Okay? And so what I'll argue is that instead of trying to predict your likelihood of getting cancer or diabetes, what we should be doing is classifying each of us into very high or intermediate or low risk, so that we can pay attention to those things that are going to affect us as we get older. Okay? So that's the objective, is to change personal health behaviors by enabling us to understand more about our own personal biology. Okay. So where we're starting on this is this Center for Health Discovery and Wellbeing cohort, or CHDWB cohort, which is a joint initiative of Emory University and Georgia Tech in Atlanta, Georgia. So the cohort is fairly small by human genetic standards. But what is interesting about it is that it's longitudinal. So there are 700 Emory employees between the ages of 25 and 75. Every 6 to 12 months, we come in and get a complete half-day medical profile, where our blood is taken, we have psychological evaluation, cardiovascular, heart tests, bone density. I don't recommend that. That's kind of scary if you don't want to see all of the fat and bone stuff all over your body when you get to my age. And then people are partnered with someone called a health coach or a health partner. So it's an experienced, so like a nurse practitioner, if you like, someone who can help interpret this information. You know, right now, clinical geneticists work with families in family planning and trying to assess what the impact of rare variants is in families. In the future, we're thinking that there will be genomic counselors or health partners who can help you interpret your clinical data and your genomic data in ways that your doctor maybe cannot do because it takes too much information. And what we're doing is genomic profiling of this cohort. And for example, we're getting complete genome sequences, complete transcriptome by RNA sequencing, methylome, microRNA, metabolome. Eventually we'd like to do all of these people and integrate that into a big picture of what's going on in terms of their functional genomics and not just their phenotype. So you might ask yourself, well, how much do people interact with their health partners? And it's a little bit astonishing to me that in fact people are very keen on that. So the average time between contact between a participant and their health partner is in fact 2 weeks to 1 month. Okay? So people actually do take use of this. And then you get the professors and other busy people who don't actually interact at all. Okay? But the question is whether or not these people respond better. So what we want is to see changes in your health risks. Okay? So there's something called the Framingham risk scores for diabetes and cardiovascular. So these are classical clinical risk scores based on obesity and blood pressure and so forth. So there are people who come into the study at enrollment who have a 30 or 40% likelihood of developing diabetes in the next 8 years. So that's really high risk. So what you'd like to see is that risk drops to the population average at least as a result of participation in a cohort like this. So a few $1,000s a year is actually very cost effective with respect to the entire medical system if this was to work. What this axis is the change in risk over the first year of participation in the study. And it's showing you that all of these people in green have changed their risk. So they're doing what you'd hope would happen. Whereas these people are not changing their risk. Now, is that because they don't pay attention to their own health? Or is it because there's something about their genetics which makes it much more difficult for their risk scores to change? So that's one of the questions that we have. People being people, actually, it turns out that probably only 10 or 15% of the people in the cohort are really responding in a positive way. Okay? And some of those will regress back to normality after 5 or 6 years. And that's universally one of the issues that we face in any sort of clinical intervention program is getting people to actually pay attention. But it's about changing the health culture. It's getting us involved more in our own health decision making. So how stable are people's phenotypes? So what these plots are doing is showing what happens to people's traits over 3 years of participation. So this is body mass index, so the ratio of your weight to your height. These people here are very healthy. They start out healthy. Maybe this one's getting a little bit less. So each point is 12 months later. These people here have actually lost about 10 to 15 kilograms. So their body mass has gone right down. You'd hope to see that happen. Blood pressure fluctuates somewhat, but in fact more people go down than up. This person's had a dramatic drop in their blood pressure. I'll come back to her later. Bone density does not change much. Serum triglycerides fluctuate. But most people actually have a value and they stay pretty much around that value for most of their adult life. That's genetics plus their environmental input as they're growing up. The question is, what can we do to bring this down to here or this down to here? And how much is that in people's own control? Well, what about genetic risk scores? Well, the simplest way to create a genetic risk score is what's called an allelic sum. So you just add up the number of, say, increasing alleles for all of the significant single nucleotide polymorphisms, or SNPs, that are found to be associated with that trait. So this plot shows actually the number of teeth in newborn babies. So there are, in fact, 8 genes which are known to affect the number of teeth. Children born with, there are 4 genes known. Children born with 8 increasing alleles actually have something like 1 extra tooth on average than children at the other end of the spectrum. Okay? So it's not a lot. It's not predictive, but it's classifying in a sense. So we can use these scores to actually estimate what the impact is. Okay? So tooth number is maybe not very clinically significant. But can we improve that? Well, it actually makes sense to adjust the allelic by the allelic effect size. So if one allele increases body mass by half a kilogram, like FTO gene does, and the next one only increases by 50 grams, then it would make sense to adjust the risk score by that. So we can do that. It turns out that does not make much difference. But also we need to weight by the allele frequency. So if everyone in the population has a tall allele, then that's not going to contribute much to the variation. Okay? But that crucially means adjusting for the population. Okay? So allele frequencies vary between Turks and Greeks and Italians and Englishmen. We have to adjust for that. So eventually genomic medicine has to be population specific. As we generate these risk scores, they have to be dependent upon the local population. Okay? So how does this work in practice? So the top 2 figures are looking at the effect of the number of variants on how much of variation we explain for different traits. So if I take the 50 SNPs that explain most of height, then it turns out that's just as good as if I take the top 150. Same with BMI. Once I get up to about 50 SNPs, I've explained quite a bit of the variation. It's only 5 or 10%, which I'll come back to that. Okay? But as you increase the number of SNPs, you increase how much of the variation in the trait you're explaining. if you actually look at it, so each triangle here is the predicted height for a person, for a man in our study, against their actual height. So what you can actually see is that the people who have more tall alleles are on average 20 centimeters taller than the people at the other end of the spectrum. Okay? And then there are always outliers. These all happen to be quite old Caucasian men in this study. We've got something in common. They're taller than they should be. And of course, you can do it for clinically relevant traits. So this is, in fact, a genetic predictor of heart disease against that Framingham heart disease risk score. Okay? It's subtle. It is not predictive, but it does classify. So these people are at high risk, these people are at low risk, and that's the information that I think we want to have. Okay? So that's where we're headed. We're not there yet, but that's what we want to be able to do. Don't read this whole figure. This actually came out in Science yesterday, an article called Depersonalized Medicine, that begins to address this question of how do we go from the population to n equals 1, to you yourself. And this is what's crucial. We need to complement the power of genomics and emphasize personal attributes of patients and their environment, and incorporate those into an enriched approach to personalized medicine. So when you go to a doctor, your doctor knows a whole lot about you. They know what your body mass is. They know how your blood enzymes fluctuate. And they interpret that when they give you a clinical evaluation. What we want to see is the same thing for genomic medicine. That you don't just send your DNA to a company and they send you a report back that says, oh, you're going to die of a heart attack when you're 55, but rather we have a profile that your doctor can work with you on to understand your health risks. Okay. Well, how important is the environment? I'm going to change gears now quite a bit and talk about a different study where we ask the question, what's the impact of environment on people's gene expression? So this was work done by a graduate student, Youssef Idafdour, from Morocco. Okay? So what we did was to go to the desert and take samples from desert nomads, from about 30 desert nomads in the Sahara Desert in Morocco. We also went to the low-income slum neighborhoods in Agadir, this city in southern Morocco. And we also went to a remote rural village in the Atlas Mountains in Morocco, and asked the question, how different are their blood profiles among each other? Okay? So we were thinking that we might find 30 or 40 genes that were differentially expressed according to those 3 different lifestyles. What we actually found was that 1/3 of the transcriptome, so 5,000 genes, were differentially expressed between people according to whether they were Berbers, nomads in the desert, or they were living in the city, or they were living in a remote village. So this is what's called a heat map. So each column is one person's profile, and each row is gene expression for a different gene. Okay? So if it's red, it means high expression. If it's blue, it means low expression. So you can see that the villagers all have a very different profile for many of their genes than the Bedouin in nomads and the people from Agadir. You see a subgroup here in the Bedouin. That's in fact women versus men. So lifestyle is a much, much bigger factor than gender in this case. Then we went back and repeated the experiment, also including Arab ethnicity. So Morocco as a country is about 1/2 Arab, 1/2 Berber. The Arabs settled in the Middle Ages, so about the 12th, 13th century. And they tend to be in the low valleys, whereas the Berbers are up in remote villages. So we sampled again, now 50 people from each site. We did these Illumina gene expression microarrays with 48,000 probes, and we measured their genotypes at 620,000 SNPs. And then we did this genome-wide association studies to find out how genotypes affect transcription. So it turns out that we were able to replicate the initial study. So this is a Venn diagram showing that there were, in fact, 7,000 genes different between the remote village and the Arab village, and 7,000 genes different between the remote village and the city, and there was a lot of overlap between that. Whereas the city and the Arab village were more similar. Okay? If you just look in the city, ethnicity and gender have no impact, essentially. But if you look in all of southern Morocco, location, ethnicity, gender, interaction terms explain quite a lot of the gene expression variation. In other words, cultural factors are having a huge impact on how our gene expression is used in our blood. Okay? And presumably that then impacts how our immune system is functioning. So in other words, genetically similar people are nevertheless phenotypically extremely different according to their lifestyle. This is a Manhattan plot because it looks like Manhattan with all of these skyscrapers. Okay? What it is a plot walking along the genome, so chromosome 1, 2, 3, all the way down to chromosome 23. These are individual SNPs, and this is a significance level. It's called the negative logarithm of the p-value. Think of this as 10 to the minus 10 significance, so 0.000000001 significance, so highly significant. This is even more so. That's what we need to be confident in genome-wide association studies. Okay? So these are each of 1,400 places in the genome where a SNP is associated with expression of the target gene. So in other words, where that genetic variant is influencing the expression of the gene that's being measured. So the next question we can ask is, where are they? And this is a plot of the location of the SNP in the genome, from chromosome 1 to chromosome 22, against the location of the target gene, so the transcript that's being affected. And 95% of them, or 99%, are along the diagonal. In fact, if we homed in, we would see that the SNP is in the same gene. We call that a local association. So in this relatively small experiment of 200 people, we can actually measure genetic SNPs that affect gene expression highly significantly. Okay? And now we can ask whether the environment influences that association. So the next thing we can do is, for each one of these cases, we can ask if there's an interaction. So this is a plot. These are all people who are AA at this SNP. These are heterozygotes. These are GG. So this is the more common variant. The blue line is the Berber village. The green line is Agadir. And the red line is the Arab village. So in all three locations, the effect of the genotype is the same. There's no interaction. This is very surprising to me. I thought there'd be a lot of interaction. So what we were hoping to see is that the green line would go that way, and the red line maybe no effect, so that the genotype effect was dependent on the environment. It turns out that's just not the case. I could show you 400 pictures like this, which say that the genotype effect is independent of the environment effect. What are the consequences of that? Let's suppose that low expression of this gene actually predisposes you to some disease, like arthritis, for example. Then which people are most at risk? It's the people in this sector down here who have the lowest expression. Okay? So they're the AA homozygotes in that particular village. All right? So even though there's no interaction between genotype and gene expression, you can see how you can very quickly lead to interaction between genotype and disease status because the gene expression itself is affected by the environment. Okay? So what I'm trying to argue for you is that it's not sufficient just to measure people's genotypes and see how they associate with your risk of disease, but we also need to include functional genomics, measuring gene expression, metabolites, protein abundance, and so forth. So does the environment matter? Well, in fact, there's little evidence for epistasis or genotype by environment from genome-wide association studies. Okay? But that's because each individual site is contributing so little to the trait. Add them all up, and I think actually we would see that the variants could collectively have a large effect on individuals. What happens is that, well, this is a technical argument I won't go into today, but it does continue to raise this central problem in predictive health. How do we go from discovery genetics to N=1 genetics? They're very different beasts. And I actually suspect that we've got our conception of environment wrong. Okay? It's not so much whether you live in Istanbul or Izmir, whether you smoke or drink, whether you're a vegan or a meat lover. So those things matter, as I've shown you. But also we need to be able to account for very individualized triggers, whether or not you were in a car accident when you were a child, whether you lost your source of income, what your parents ate when you were 2 years old. These sorts of things are very individualized and cannot be captured by epidemiology, and yet need to be captured by predictive health genetics. So this is my group in Atlanta. This is my lab manager, Dalia. If you see her in Istanbul in a couple of weeks' time, be nice to her. It's her honeymoon, okay? You can see that most of you would fit in well with the phenotype in my group. So this is another way of portraying what I was just saying. Imagine a metabolic pathway. This is simple central metabolism. We have variation in the activity of each of these enzymes, which we can represent by red and green and yellow. So high activity, low activity. That's due to structural variants that affect the protein function, and it's due to regulatory variants that affect how much of the enzyme there is. As a consequence of that, two different people will actually have different, what we call metabolic plaques. They'll have different levels of activity through that, and that might impact their likelihood of being obese. Okay? So this person has relatively low activity, this person has relatively high activity, but evolutionary theory actually tells us that the system will evolve so that both of these are consistent with health. Now the question is, what happens if we mutate a single gene? In this person, it doesn't matter. They just reduce from here to here. But in that person, that same mutation has a really big impact. It's what we call penetrance phenotypes. So that's our model for how common variants and rare variants interact to determine the personal genetic risk profile. So I'm going to skip over this slide. Well, actually, how much longer have I got? How are we doing on time? 50? Okay. So, I mentioned one thing we're doing is measuring people's gene expression in the whole cohort long. The first visit, second visit, third visit. This is just showing 12 people, how common, how persistent their gene expression is. Okay? So we're averaging the expression of 15,000 genes. And we're looking to see how similar that is between first, second, and third visit for 12 people. And the answer is that those 12 people are actually very consistent. This is one person, second person, third person, fourth person. So people's gene expression profiles don't change much. There's just one person here who's changed a lot. Their methylation profile also changes. So something's different about that person. But most people, they stay the same. So what we've been doing, I will go through this quickly, is actually if you profile gene expression, we can identify, it's actually 9 now, different, what we call axes or modules of variation. So all of these genes are co-expressed in people. These genes are co-expressed in people. And it turns out that these axes capture T cell function, platelet and red blood cell function, B cell function, inflammatory signaling, interferon response, viral response, metabolic activity. Our gene expression profiles are very, very highly structured, it turns out. So we think that we can, by measuring gene expression profiles, potentially define people with respect to their susceptibility for immunodeficiencies, blood disorders, inflammatory diseases, and viral responsiveness. That's our hypothesis. We have to prove that by measuring clinical measures with gene expression in tens of thousands of people. And that's what we're working towards doing, and then asking if that improves on genotypes. By the way, this is work done by an undergraduate who was working in my group, Marcelo Prani. So you may have heard of a man by the name of Mike Snyder. He's a professor at Stanford who measured his blood every 2 weeks for 2 years. He sequenced his own genome. He sequenced his RNA every 2 weeks. He measured his proteins every 2 weeks. He measured his antibodies every 2 weeks. He did everything under the sun possible every 2 weeks and published a paper on it. Okay? It's called the Snyderome. Okay? What happened is his genome told him that he was at high risk with diabetes. There was no diabetes in his family, and yet his genome said that he could be at high risk of diabetes. Soon after entering the study, he got a respiratory virus infection. A respiratory virus. He could not breathe because of viral infection. 6 months later, he had a second viral infection, after which his blood glucose levels started going. He became pre-diabetic. So when he talks about this, he says this is purely predictive health genomics, that his genome predicted something would happen. What we're showing here is that our axes of variation actually track much of this, what's happening. So when he gets a viral infection, this axis of blood goes whoop. Here's his second viral infection. The next day, whoop. Okay? So this set of 200 or 300 genes mark that incidence of viral infection just for a few days, transiently. Okay? And in fact, that's highly enriched for interferon response. When he gets diabetes, which is around about here, this axis and that axis and that axis all change quite dramatically. Okay? So even though people's profiles are fairly constant over time, we actually think that this technology could help us to actually track what's happening as we're responding to viral infections, or as we get a serious chronic illness. You can imagine if you're recovering from cancer, doing this sort of profiling to see how your immune system is responding to drug treatment. So that's one direction we're going. The next thing we can do is, I skipped over this, but basically what we're doing is asking which genes are differentially expressed uniquely for each person. So all of us have about 30 or 40 genes which are just barely being expressed compared to everyone else. That's like having a mutation in that gene. Okay? And now we can ask, do they have something in common? Collectively, is that going to change our risk? So here are 2 examples of personalized genomics. So this individual is a 44-year-old white male, not me. Okay? He has high levels of expression of this gene, which is known to be associated with leukemia. He has low level expression of this gene, which is known to be associated with leukemia. Same for that one. Same for that one. And he has the lowest level of B cell signaling in the entire study. I would want to know that if it was me. That's what we're talking about using predictive genomics from this. He's got at least 5 markers of leukemia in his blood. He actually is now receiving treatment for cancer. But I don't know what type of cancer. It may not be leukemia. But it's just an example of where we want to go with functional profile. Here's another person, a 63-year-old African-American woman. It's a very strange phenotype. She's got extremely high body mass, over 35, extremely high body fat, but extremely low glucose and extremely low triglycerides or fats. Those things don't normally go together. What's going on with her? And she's not diabetic. She ought to be diabetic for a lot of reasons. She's also got renal tubular acidosis, a history of kidney infection, which are known to be associated with mutations in those genes. Possibly the low glucose relates to not expressing this enzyme. She's got a very low inflammatory response, and she has very high levels of this hormone, which is probably protecting her to some extent from diabetes. Now the problem is with all of this that it's anecdotal, okay, at this point in time. I can't prove that any of those gene expression levels are causing the particular phenotypes. But that's the challenge of going to N=1 genomics. How do we use our functional information to enhance our genotypes, to better understand our own personal health risks? Okay? So what we're working towards is that people will get profiles of their genetic risk on this axis and their clinical risk, so by measuring their actual clinical parameters with respect to immune function, metabolic function, heart function, musculoskeletal function, respiration, psychology, cognition, cancer, reproductive health, and so forth. So that's our model, is that people will get genomic profiles, compare them to their clinical profiles, and see whether they're concordant or discordant. So this person has very high genotypic and clinical risk for heart disease. That's what they should focus their behaviors on. Whereas their cognitive domain is actually very little risk. So maybe they don't focus on that. And then what you can do, once you know that your health is in a certain area, focus on it. So, just two examples. This was the person in the beginning who had very high blood pressure. So these markers here are showing high blood pressure. She has 0 predicted risk for blood pressure. So maybe knowing that you don't have a genotypic risk might convince you that maybe you've got some environmental or some dietary responsibility and you can actually do something about your blood pressure. On the other hand, these two people up here, what these markers are showing have no risk of diabetes, no signs clinically of diabetes, and yet genotypically they're at very high risk of diabetes. So they may say, look, I'm fine now, but I need to be aware that maybe 5 or 10 years in the future I'm at high risk, and then pay more attention to that domain. So that's what we like to see about people changing their health behaviors based on their genomic understanding. Just in the last 5 minutes, I'm going to switch gears one more time and just leave you with a more conceptual idea. This sentence is taken from a book I wrote a few years ago called It Takes a Genome, which hopefully will be available in Turkish very shortly. Thanks to Professor Ergi, who's translating it into Turkish. I very much appreciate it for that. What I'm arguing in this book is that there's a disequilibrium between our genotypes and our environment. And because of that, we're at high risk of certain diseases. So globally, what's happened is that we're moving from diseases of poverty in low-income countries, largely infectious diseases, to diseases of the West all over the world. So in fact, the World Health Organization estimates that in just about all countries, cancer, diabetes, asthma, depression, and Alzheimer's will be the major sources of morbidity in the next 10 or 20 years. So why is that? What's going on? So partly, obviously, it's because we're cleaning up our environment, but that doesn't explain why disease risk is increasing. Okay. So how do we think about disease risk and genetics? Well, we can, as I've shown you, we can actually add up the number of susceptibility alleles for any one of hundreds of diseases, and they will be normally distributed. And evolution has ensured that very few people are above a threshold of something, so they're not going to get disease. Maybe 1% of people get disease. In the last 2 or 3 generations, that profile has changed completely, so that now 5 or 10% of people get disease. And the way most people think about that is as a shift of the entire susceptibility spectrum to the right. Well, of course, genotype frequencies do not change quickly. So it's what they're doing that's changing. Another way to think about this is what's called decanalization. So it's not so much that the distribution is shifted, but that it's broadened. Let me introduce my two Shelties. Okay, these are my dogs on a beach in North Carolina. This is Namzara, or Princess Namzara, and this is Lady Brisbane or Brizzy. Okay? I want to say three things about these two dogs. They are twin sisters, same liver. Okay? She is half the size of her. Zara's the runt of the litter. She's not here, I can say that. Okay? She's half the size, and yet she's completely normal. In fact, she's very spunky, she's very bright, very active. How does development allow us to buffer things so well that you can end up half the size and be completely normal? Biological systems are very, very good at buffering. Let's focus on Brizzy now. This ear here is floppy. Shelties are supposed to have two floppy ears. She only has one. So the two sides of her body are different. We also have to be able to do that so that our legs are the same length. That happens. Fluctuating asymmetry, which is a technical term for this, is a very important concept in understanding robustness of the system. You cannot see it, but if you count her whiskers, she has 17 whiskers on each side of her snout. And you're probably thinking, why am I counting my dog's whiskers? Right? Some of you will go home and count your dog's whiskers, and I guarantee that you will find they have 17 or 18. Well, I hope. Mice always have 17 or 18 whiskers on each side of the snout. Always. Except in the presence of a mutation called tabby. So tabby mutant mice have 12 whiskers on average. But the interesting thing is they have anywhere from 5 to 20. Okay? So the wild type state is you always have 17 or 18. In the mutant state, you have anywhere from 5 to 20. What that tells evolutionary geneticists is that biological systems evolve to be more stable. They reduce the variability. So the idea is that in modern human society, we've evolved rapidly as a species, and we've changed our environment, and we're destabilizing those systems and leading to more risk of disease. And I'll just show that. So how might that happen? So one possibility, let's imagine the size of your liver. Okay? What can determine the size of the liver? Well, it's the number of cells on this axis, cell number, and the size of the cells. So there will be some relationship between those two things. And usually it will not be linear. So what evolution does is it finds an optimal size for your liver, which is the purple or magenta line here. The population evolves to that line, but it does not stop when it gets to the optimum. That's what we usually teach in evolutionary genetics, is you evolve to the optimum. In fact, it will keep evolving to the point where the population is most robust. That's here in the middle, because a small change there can be compensated by a small change in the opposite direction. Little changes, little compensation. If you're out here, same change, small change requires a big compensation. Same at the other end. So the system is most stable here in the middle. Alright? So let's think about diabetes. So one way of thinking about this is what we have to do is maintain our blood glucose levels. That's essentially what goes awry. Okay? So if the population's here, small changes, we all stay. As you increase your insulin resistance and insulin release, eventually you get to a point where that can no longer be sustained, and you fall into impaired glucose tolerance and eventually diabetes. Well, if the population has moved along this axis, then more people are at risk of falling off that. So that's one way of thinking about what's happened, what's the relationship between human history, population history, and increasing risk of disease in modern society. And we can do similar strategies for just about any complex disease that we care to think about. And so I just leave you with that thought about the complexity of the relationship between genetics and disease. Okay? So just summarizing, GWAS typically only discover 5 or 10% of disease risk, I didn't tell you this, but it actually turns out the statistical methods allow us to infer that in fact at least 50-75% of the risk is due to common genotypes. So all diseases are incredibly genetically complex. Now rare coding variants are all the rage, particularly in psychiatric and craniofacial and immunogenetics right now, for good reason. They're clinically very, very useful for a lot of children in particular. We're arguing that extreme expression values may turn out to be just as useful. But I also said that we're not yet sure how to generate these profiles and turn them into ones that are clinically better than family history or classical biomarkers. That's where the field is trying to get to. And our notion then is that common variation together with the environment regulate genes in pathways, and that people can be classified with respect to risk by understanding how those pathways are co-regulated, and then how rare variants act on top of that to push us outside of these buffering zones into disease. And if that's all true, then personal health choices maybe can use to modify our profiles personally, and the functional genomic profiling will be a part of personalized medicine in the future. And with that, I'll stop and take any questions. Thank you. Questions? English or Turkish? Maybe I should ask the question. Please.
Thank you for your nice presentation. I would like to ask, did you face any ethical problems while you are doing this experiment? Because knowing or not knowing of people's genetic risks of diseases are quite an ethical issue, I think.
That's a really wonderful question. Thank you very much for asking. So we are not currently allowed to give genotypes back, genetic information back to people. So there's a bit of a double standard, because we give people clinical data and trust them with that data, and they make decisions, but we cannot give the genetic data. And a part of that is because people are afraid of genetic data. What I hope to do this summer is actually do a pilot study where we would give people false information, but not deliberately. We would tell them that it's false and try to ascertain from them, try to have them tell us what they want or what they don't want. Okay? So we think that there will be some people who want this information, some people who are not sure, and some people who definitely do not want it. For example, I had a student who bought 23andMe for his father. He didn't want it. Gave it to his mother. She didn't want it. Gave it to his girlfriend. She didn't want it. So he ended up keeping it for himself. Okay? So some people want it, some people don't. I would say, this is very important, so breast cancer, BRCA1, if you have a mutation in BRCA1, you have an 80% lifetime risk of breast cancer. It only explains 1% of all breast cancer, so most breast cancer has nothing to do with it. But those people who have that mutation are at very high risk of breast and ovarian cancer. In 23andMe, just last month, they published a study of attitudes towards finding out that. So if you do that, you have to do a tutorial to get your answer. 13 people found out that they were BRCA1 positive. What they report in this paper is that all 13 eventually found that it was a positive thing. So all of them gave that information to their family. More family people got tested. They found another 30 cases that way. These are people who are now either having surgery or taking other steps to reduce that. So people are initially very scared, but once they get over that, they were fine with it. So I think it's a really important example, but it's a great question which we'll follow up. Thank you.
your presentation. Now we will present your certificate of 2 saplings that planted in your name in the 7 trees forest. Ara vermeden önce bir duyurumuz var. Karadeniz Teknik Üniversitesi Biyoloji Bölümünün düzenlediği 8. Moleküler Biyoteknoloji Bahar Okulu 19-22 Nisan 2013 tarihinde yapılacaktır. Karadeniz Teknik Üniversitesi tüm katılımcılarımızı davet etmektedir. Şimdi 15 dakikalık kahve molası.